Hi. In this video, we'll resume our discussion of transcription. In the previous video, we had just concluded talking about transcription start and stop sites, which we now talk about in more detail. Here are detailed views of those regions. Here we have the transcription start site. That's where we're going to start making RNA. I want to mention that this is typically not the same as the translation start site, where one starts making the protein. We're going to start making the protein somewhere around here, but we're going to start making the RNA here at the +1 site. The region upstream of where the gene starts is called a promoter region. It is also called an operator region, and some people make a distinction between them, but it isn't really necessary. When they do, the operator is typically a very close region near the start site, maybe the first 100 or couple hundred base pairs, whereas a promoter can be thousands of base pairs upstream of the transcription start site. But an extremely important part of any promoter or operator is the 35 bases immediately upstream of the start site. This is called the TATAA box. The reason it's given that name is because of the preponderance of T's and A's in the area highlighted green, which are also conserved. Notice that the TATAA box has the characteristic spacing. It's 10 bases upstream of the transcription start site, and the other part starts at -35 base pairs. Now you already know that T and A bases will unzip a little bit more easily than G and C base pairs, so that may be part of it. I should add that you also know by now that it's not because there are 3 hydrogen bonds in GC base pairs and 2 hydrogen bonds in TA base pairs that makes it easier to unzip, but rather the stacking interactions between the bases. But they do unwind more easily, so it's thought that one of the reasons you have lots of A's and T's in this region is to help the region open up more easily so that the polymerase can start making the mRNA molecule. The termination or stop signal is less well characterized, but it has this characteristic run of T's right before the stop, and frequently there is a hairpin formation which will help signal the polymerase to stop transcription. Another feature about the TATAA box, which you can see here in the crystal structure of a protein which helps initiate transcription in blue and green, called the Tata Binding Protein. When this crystal structure was solved, people were rather amazed at the very significant angle of about 60 degrees at which DNA is bent. This bending ability at the sequences is a consequence of the AT-rich nature of this portion of the nucleic acid, and highlights the fact that other characteristics, rather than just hydrogen bonding possibilities in the major and minor grooves, contributes to sequence-specific DNA recognition by proteins. In this case, it is the bend of the overall structure associated with runs of A's and T's. This slide shows that genes can be coded on either strand of the genome, and that they are oriented in the same direction on that particular strand. Because genes are always coded in the orientation so that the mRNA will be synthesized in the 5' to 3' direction. Also notice that genes can in principle overlap when they're on different strands, but this is a rather rare event. This slide highlights the fact that the RNA polymerase is not a single protein, but rather a complex machine with lots of different protein parts. You won't be responsible for knowing most of these, but it's important for you to see it at least once. In general, all these proteins, TF2D, TF2B, E, and H, these are known as transcription accessory factors, or TAFs, TAFs, and they are utilized at essentially all transcription start sites. The initial binding event to start transcription is performed by the Tata Binding Protein, which is part of the TF2D complex, and it binds to the TATAA box on the DNA. This helps the recruitment of other factors like TF2B, the RNA polymerase complex, and other factors. When this complex gets assembled at the transcription start site, then transcription can begin. The book sometimes refers to these as transcription factors. Most biologists would not call them transcription factors. That's a term reserved more for proteins that bind only at a select few number of genes to turn them on specifically. The last point to make is these proteins assemble at the transcription start site, but as transcription starts, most of them will be removed from the complex and the polymerase will proceed by itself. The gene-specific factors, or true transcription factors, occur at regions called enhancers. These can be close or very far upstream from the TATAA box. In fact, it can be many thousands of base pairs away. And for a long time it was wondered about how a protein binding thousands of base pairs away could influence proteins binding here at the TATAA box. It's almost embarrassing in retrospect, but simply by the DNA looping around, these proteins can be in immediate contact with the other proteins and all contribute thermodynamically to a greater binding energy to assemble a complex at a specific location to turn this gene on and for transcription to occur. On this slide, we highlight some of the differences between prokaryotes and eukaryotic mRNA. There are at least 3 very important differences here. We'll start with the eukaryotic mRNA. First, there is this cap which is called the 7-methyl-G cap. The 7 is the location on the gene to which the methyl is added. This cap structure is important for proteins to bind, and they're also for the stability of the sequence. Also, every transcript ends with a poly-A tail of approximately 200 bases. Eukaryotic mRNAs almost exclusively code for a single protein, one of the biggest differences for prokaryotic mRNAs and eukaryotic ones. With prokaryotic mRNAs, a single mRNA transcript can code for several proteins, referred to as a group of genes in an operon. In this example, there will be 3 different translation start sites. Ribosomes will bind here, here, and here, and they'll make multiple proteins from a single mRNA transcript. These proteins are almost always functionally related. In this example, it might be that the 3 proteins are all involved for a particular type of amino acid synthesis. This is often referred to as mRNAs of bacteria, being referred to as polycistronic. This slide highlights another important difference between bacteria and eukaryotes, and that is that bacterial genes almost exclusively only have coding regions for every RNA. Here's the transcription start site, here's the translation start site, and this entire RNA stretch codes for protein. In eukaryotes, there are different regions called exons and introns. Exons code for protein. The introns are intervening material, and they have some functions, but less is known about them. The discovery of introns was performed by Richard Roberts and Philip Sharp. Dr. Roberts is an adjunct professor here at Boston University, and he received the Nobel Prize for his part in the discovery. So in eukaryotes, what will happen is the organism will transcribe the entire gene, and it will then cut out the intron regions, and you'll be left with a final transcript that is much shorter, a mature mRNA, which will be the code for the protein. The size of human proteins can vary quite a bit. Some can extend over extremely large regions, in this case shown 200,000 base pairs, and you can see that most of the sequence is in fact introns. There are examples that are even larger than this and extend over 1 million base pairs. One of the potential advantages of using many exons to code for the protein is that you can incorporate some modularity, and these transcripts can be alternatively spliced. That means in different situations you can utilize different exons, even though all of them are initially transcribed. You can combine them in different combinations to make many different types of protein from a single RNA transcript. There are many different kinds of logic that can be incorporated here, and just about anything that you can think of you can find an example of. In this part, exons 2 or 3 are used in an either-or fashion. Also, there are multiple final exons, allowing the cell to pick different carboxy ends to the gene. Also, sometimes exons are skipped altogether. So that's what I wanted to say about transcription. We'll move on to translation with the next video. Thank you for listening.